I think we have waited enough. I still people will join on the way, but yeah, I think we should start now. So, good morning everyone and welcome to the CCEMP seminar series. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owner of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the lands from which everyone is joining us today and pay my respects to elders past, present and future. My name is Inamur Rahman and it's my pleasure to chair today's seminar. Just a few housekeeping points before we begin. Please keep your microphones muted during the presentation. We will have questions at the end and those joining via Zoom can enter their questions in the chat which I will read out during the Q&A. The seminar is being recorded for your information and the recording will be shared through the CCMP website. It is my pleasure to introduce Dr. Melanie Dietrich from the Walter and Elizabeth Hall Institute. Melanie is an structural biologist interested in host pathogen interactions and antibody-mediated inhibition of infection. Her research combines structural and biophysical approaches, including cryo-EM and X-ray crystallography, to understand malaria parasite biology and develop transmission blocking strategies. Today, she will talk about a novel transmission blocking vaccine candidate against malaria parasite, focusing on the Plasmodium falciparum 230 and Plasmodium falciparum 48-45 complex and its potential as a target for malaria transmission blocking vaccines. So without further delay, please join me in welcoming Dr. Melanie Dietrich for her exciting talk. Over to you, Dr. Melanie.
Thank you so much for the kind introduction. I hope you can see my slide.
Yeah.
Oh, okay. Yeah, good morning and thanks everyone for joining today's seminar. My name is Melanie Dietrich and I'm a postdoc in the Tham lab at WeHi, and today's talk will focus on two proteins of malaria parasites that play a critical role in transmission. So in the Tham lab, we work on different pathogens, malaria parasites, but also coronaviruses and other pathogens with pandemic potential. We develop antibody-based interventions and mRNA-LNPs and use structural biology for rational target design and to understand how our antibodies and nanobodies bind their targets and how they function. So malaria remains a devastating disease with over 600,000 deaths in 2024. There are currently two vaccines available, termed RTSS and R21 matrix M, but they only provide partial and short-lived protection. The life cycle of the parasite is quite complex and involves two hosts, a female Anopheles mosquito and humans. And as the parasite adapted to survive in two different hosts, they change into several distinct forms as it moves between the two hosts. So the WHO, and I guess it's widely accepted now, that no single tool will solve the problem of malaria, and that really multi-stage strategies are needed to stop infection and transmission. So the focus of my research for probably the past 7 years have been the existing proteins of Plasmodium. Sorry, I have to move this bar out of my way. Okay, sorry. Yeah, these proteins form a small protein family of 14 identified members, and they are conserved across Plasmodium species. They are among the most abundant surface antigens, and they are present on all developmental stages of the parasite. These members only share a low amino acid sequence identity among each other, but they have a common structural feature, which is the 6-cis domain. Members have between 2 but up to 14 of these 6-cis domains. And as the name implies, these domains have up to 6 cysteines that form disulfide bonds. The 6-cis domain is a fairly small domain of around 120 amino acids that fold into a beta sandwich, and up to 3 disulfide bonds stabilize the domain. The 6-cis domain is structurally related to the SIG1-related sequence domain in Toxoplasma, which members play roles in cell attachment and immune evasion. And these two domains are suggested to have evolved from a common ephrin-like ancestor. Some members of this existing protein family have been implicated to play really critical roles in the parasite life cycle, and some are found as heterodimers on the parasite surface, such as P12 and 41, 52 and 36, and 48, 45, and 230. In all these 3 heterodimers, one of the interaction partners has a GPI anchor, these ones on top, and the other ones not. Host cell interaction partners have been identified for some of the existing proteins, such as for sporozoid proteins 52 and 36. They play critical roles in liver cell invasion through the interaction with ephrin receptor A2, CD81, and SRB1. P92 is a blood stage parasite protein and plays a role in immune evasion through its interaction with factor H. And P47 is a mosquito stage protein that plays a role in mosquito host tropism through its interaction with P47 receptor. And 48, 45, and 230 are present on sexual stage parasites and critical for parasite fertilization, and these proteins are the focus of my talk. Fertilization of the parasite actually occurs in the mosquito midgut. So when a female Anopheles mosquito takes a blood meal from an infected human, it takes up male and female gametocytes that quickly mature to male and female gametes that fertilize and form a zygote that develops into a mobile ookinete that traverses through the midgut epithelium and embeds itself at the basal side of the midgut wall as an oocyst. Within the oocyst, thousands of sporozoids develop that, upon rupture, travel to the salivary glands of the mosquito, where they are basically ready to infect the next human during the next mosquito bite. So just keep the oocyst in mind, because we'll come back to it in a bit as we use them as the readout in our transmission blocking assays. Currently, there are only a few proteins known to play a role in fertilization, and among them 48, 45, and 230. So here's the whole parasite life cycle depicted, and fertilization is an obligate step of the parasite life cycle. So when we are able to block fertilization, we prevent onwards development of the parasite in the mosquito, and thereby prevent transmission of the parasite to new human hosts. So why do we want to target parasite fertilization? It's because the blood stages of infection, which are responsible for all symptoms of the disease, have really high parasite numbers within the billions. So in comparison, the number of parasites significantly drop in a mosquito, where usually just a handful of parasites develop into oocysts. And this represents a significant bottleneck in the parasite life cycle, and means that we only have to target a small number of parasites. Also, the targeted antigens are less polymorphic than antigens of the blood stages. There are different transmission blocking interventions under development, such as recombinant vaccines. They work by immunizing a human where malaria transmission occurs that induce antibodies against mosquito stage antigens. So when the mosquito takes a blood meal from a human, it takes up gametocytes and also these antibodies that when the parasite emerges from the surrounding red blood cells in the mosquito midgut, these antibodies can bind and prevent further development in the mosquito and prevent onwards transmission. So these vaccines would not prevent infection of the vaccinated human, but prevent onwards transmission. Currently, there are a number of recombinant vaccines targeting fertilization antigens 48, 45, and 230 under development, and some of them have already reached clinical trials. Then another approach is basically passive immunization, where monoclonal antibodies are directly administered. Monoclonal antibodies have the advantage that they are highly specific and can take basically effect immediately, and they do not rely on a functional immune system. So currently, there are only a few monoclonal antibodies licensed for infectious diseases, but many are in development, including 5 in clinical trials for malaria, of which 1 is for transmission blocking. So these interventions have particular application for seasonal deployment in high transmission areas or high risk groups, and for preventing the spread of resistant strains and targeting outbreaks in areas nearing elimination. One second, please. Excuse me. In addition, bed nets, drugs, and diagnostics are additional important tools used to reduce transmission. And we are aiming to expand the repertoire of transmission blocking interventions further with nanobody Fcs, bispecific nanobodies, and mRNA-LNP vaccines. So what are nanobodies? Nanobodies are the antigen-binding regions of so-called heavy chain-only antibodies, which can be found in camelids like alpacas, llamas, and camels. And these nanobodies are great because they are small, they are robust, and can be easily linked together to form different antibody formats. So we have generated nanobodies against a whole range of different sexual stage proteins to block transmission of the parasite. And therefore, we use the WIHA nanobody platform. Among them, we of course generated nanobodies against 230 and 48, 45. So 230 and 48, 45 are present on both male and female parasites. And knockout studies have shown that they are important for male fertility. And these proteins are leading transmission blocking vaccine candidates for over 30 years now, and they belong to the 6-cysteine protein family, where 230 is the largest member. It has 14 6-cysteine domains, and it also has a very long N-terminal unstructured region that gets proteolytically cleaved during gamete maturation. 230 is present on the parasite surface via its interaction with 48, 45, which has a GPI anchor and 3 6-cysteine domains. Transmission blocking vaccine programs mostly focus on domain 1 of 230 or domain 3 of 48, 45. And crystal structures were available for the first 2 6-cysteine domains of 230 and the whole ectodomain of 48, 45. It was shown that human monoclonal antibodies, quite a few have been identified against these proteins that are able to block transmission. So to test if our nanobodies are also effective in blocking transmission and to better compare them with known transmission blocking monoclonals, we have used our nanobodies to the Fc region of human IgG1. And we used a Wehi insectary where we feed female Anopheles mosquitoes a blood meal containing sexual stage parasites, as shown here. So here you can see the mosquitoes feeding, and the blood meal is kept warm with warm water running through the feeders and mimicking body temperature. So 1 week later, we dissect the mosquitoes and harvest the midguts. And here you can see stained midguts of an infected mosquito, which shows these little red dots, which are the oocysts of the parasite. So these assays are called standard membrane feeding assays. And to test our nanobodies, if they have an effect in reducing oocysts, we add them and control antibodies also to the blood meal containing the parasites. So we generated nanobodies against a 230 construct that contained the first 2 6 domains, and by using crystallography, you were able to show where our nanobodies bind, and 2 of them bound to different epitopes, but both within domain 1 of 230. In our transmission blocking assays, we found that when we dissected the mosquitoes and counted the oocysts, we found that compared to wild-type parasites or in presence of a non-relevant nanobody control, our nanobodies termed F5 and F10 were able to significantly reduce the oocyst burden. A cocktail of the 2 nanobody Fcs had no further improvement, and unfortunately our nanobodies were not as efficient as LM4230, which is a known human transmission blocking antibody against domain 1 of 230. Similarly, for our 48-45 nanobodies, we generated nanobodies against domain 3 of 48-45 that recognize epitope different to TB31F, which is the most potent known transmission blocking antibody to date. Our nanobodies recognize their antigen with high affinity, and as shown in these IFAs, they recognize the protein on parasite material. In our standard membrane feeding assays, the nanobodies significantly reduced the oocyst number as compared to wild-type control and non-relevant nanobody control, but not as efficient as TB31F. This study was led by Frankie as part of her PhD and was recently published in PLOS Pathogens. Frankie also generated bispecific nanobodies, where 2 nanobodies that target 2 different antigens were fused together and in the Fc format. And Frankie found that compared to the original nanobodies that either recognized 230 or only 48-45, the bispecifics were able to recognize both 48-45 and 230 with high affinity. And they were also able to reduce oocyst numbers significantly, demonstrating that our nanobodies are a versatile antibody format. Then an exciting approach of our nanobodies with translational potential is currently developed by our collaboration partner Dina at Imperial College London under the wider Transmission Zero program, which aims of propagating malaria-resistant mosquito populations. So Dina developed develops transgenic mosquitoes that express nanobodies tissue specifically. So either in the cardia and anterior midgut or the posterior midgut. And here as an example, she uses nanobody F10 or nanobody F10, which targets domain 1 of 230. She uses PCR to confirm integration of the nanobody into the mosquito genome, and QRT PCR and Western blotting to validate nanobody expression, as well as IFA, where you can see here that our nanobody F10 was expressed tissue specifically to the posterior midgut. In an initial experiment, Dina infected the parental parasites as well as parental mosquitoes, as well as mosquitoes expressing F5 or F10 with sexual stage parasites, and found that ookinete invasion was inhibited in the mosquitoes expressing our nanobodies, as shown by the reduced relative expression of Pfs1. This is an enzyme which is really crucial for the ookinete to traverse the midgut epithelium. And this indicates that our nanobody expression effectively blocked parasite development in these mosquitoes. So far, our work focused on domain 1 of 230 and domain 3 of 48, 45, the domains that were previously shown to be targets of transmission blocking antibodies. And I hope I could show you that our nanobodies against these domains are also able to block transmission. So next, we wanted to answer a few open questions of the field. So although the two proteins have been leading transmission blocking vaccine candidates for a long time, it still remained unclear how they actually interact with each other. And we also wanted to investigate if there are other domains in 230 rather than domain 1 that may be targeted for transmission blocking antibodies. So as 230 is a fairly large protein, and both proteins are rich in disulfide bonds, which makes them challenging to produce recombinantly. That's why we generated a parasite line where 230 has a C-terminal purification tag. So we cultured these parasites, and yeah, I performed affinity and size exclusion chromatography to purify this protein from parasite material. I obtained a nice peak on size exclusion that contained 230 as well as 48, 45, and I used mass spec to validate the protein identities. You can see that some of 230 runs as a doublet, which is known, or which happens during gamete maturation, where the long N-terminal unstructured region is proteolytically cleaved. So either some of the parasites already underwent gamete activation, or in the lysate, the proteases could access to 30. So yeah, we used this nice peak for cryo-EM studies and obtained a cryo-EM map at a global resolution of 3.36 angstrom, which showed some preferred orientation bias. With the help of EM-Ready, we were able to improve the map interpretability, and with local refinement, I was able to resolve this part of the structure a bit better. So seen in this composite map, you can see that, yeah, this part of the structure has a significantly lower resolution than this part, but the interaction site of the two proteins are always present in the better resolved part of the map. I used AlphaFold and crystal structures to model the proteins into the map. And you can see the domain organization of the two proteins, where domain 1 of 230 is far away from the 48-45 interaction site. Yeah, 48-45 is in contact with mostly domains 13 and 14. But let's have a closer look. Here you can see the three exist domains of 48-45 in orange, which are contacted mostly by domain 13 and 14 of 230, and the very C-terminal end of 230 runs between domain 1 and 3 of 48-45. So if we flip the structure around, you have a better look at domain 1 of 48-45, which is surrounded by domain 14. Domain 2 of 48-45 sits on top of domain 14, and domain 3 forms interactions with both domain 13 and 14. So having established that the C-terminal domains of 230, domains 13 and 14, are involved in binding to 48-45, we generated a transgenic parasite line that lacked these two domains. These parasites were viable and expressed to 30, as you can see here in this Western blot, where the protein runs as expected to slightly lower molecular weights. This IFA shows that 230 and 48-45 are expressed in these late stage sexual forms of the parasite, but in these gametocytes, these parasites are still surrounded by a red blood cell, and therefore we can't comment if 230 is actually localized at the parasite surface in these parasites. So when we activate gametocytes to gametes that have egress from the red blood cell, you can see that in wild-type parasites, 48-45 and 230 are present on female and here male parasites, while in our transgenic parasite line, 230 is missing, which indicates that the domains 13 and 14 are critical for the localization of the protein to the parasite surface in male and female gametes. We also examined whether the transgenic parasites had a defect in parasite fertilization using the standard membrane feeding assay. And we've observed a substantial reduction of oocyst numbers in the transgenic line, indicating that the two domains are critical for the role of 230 in parasite fertilization. We next generated nanobodies against these two domains from immunized alpacas and obtained 4 nanobodies that bound with high affinities, and they fall into 3 epitope bins. To see if these nanobodies have an effect on the 48-45-230 complex, we performed a pull-down assay using a metoside lysate and the transgenic parasite line with the purification tag, actually a twin strep tag, and we used streptectin beads to pull down to 30. We use anti-strep HRP to detect 230 on these Western blots and a specific mouse monoclonal for 48-45 detection. In the input sample, you can see that both 230 and 4845 are present in the parasite live state, and unbound sample shows that all of 230 bound to the beads, while there's some 4845 which is not in complex with 230. And the elution shows that both 230 and 4845 co-precipitate. So if we have the addition of non-relevant nanobody control in our live state, we see similarly that these nanobodies have no effect on the complex, as we detect both 230 and 48, 45 in the elution. We then tested if the non-human monoclonal antibodies LM4-30 and TB31F had an effect on the complex. LM4-30, which binds domain 1 of 230, had no effect on the complex, as we observe both 230 and 48-45 in the elution, while TB31F, which binds domain 3 of 48-45, was able to disrupt the complex, as we only see 230 and not 48-45 in the elution. Similarly, for our nanobodies, we found that only 230 is present in the elution and 48-45 is missing, which indicates that also our nanobodies were able to disrupt the complex of the two proteins. So if we have a quick look where these transmission blocking antibodies and nanobodies bind, there are crystal structures available of antibody fragments of these non-human monoclonal antibodies, and we were also able to obtain crystal structures of our nanobodies in complex with domain 13-14. So when we align these crystal structures onto our cryo-EM structure, you can see that LM4 to 30 binds domain 1 and far away from the 48-45 interaction site, and didn't show or had no effect on the complex, while TB31F recognizes domain 3 of 48-45, which is shown in the side view, and would sterically clash with 230 and therefore compete for binding to its antigen. For our nanobodies, two of them bind to domain 13 of 230, and they would clash with other domains of 230. So at the moment, we think that they the nanobody binding would cause a conformational change in 230, which affects 230's binding to 48-45. But this needs further investigation. And our third nanobody binds to domain 14, mostly of 230, and it binds where usually domain 1 of 48-45 binds to 230. And this nanobody would directly clash with 48-45 and compete for binding to 230. So next we wanted to see if other domains of 230 other than domain 1 are targets of naturally acquired antibodies. And therefore Ivo Müller and Ria Longley from WeHi performed a multiplex assay using serum samples from children in PNG, where Plasmodium, falciparum, and Vivax are hyperendemic, and they screened for reactivity to different recombinant proteins. Their cohort had children with current Plasmodium infection or children with currently negative but prior infection, and they used malaria naive individuals from Melbourne as a control. So we tested 4 different constructs of 230, one comprising domain 1 and 2, one 5 and 6, one D11, D12, and 13 and 14. And we found that, individuals living in malaria endemic areas can develop immune responses to all of these 230 constructs through natural exposure to the parasite. The mean IgG levels against the constructs were the highest in individuals with current infection. So then we developed mRNA-LNPs against domain 13 and 14 of 230. And therefore we used 2 different constructs, one termed long and one termed short. And they both contain the 2 6 domains, the 2 C-terminal 6 domains. And the long construct has also the very C-terminal end of 230. While the short construct, these 24 amino acids are missing in the short construct. We did 2 immunizations. So we immunized mice on day 1 and day 28, and took 2 bleeds, one at day 21 and one at 68, and found that the second immunization of both constructs resulted in a significant boosting of antibody titers. And see here. So sera of these immunized mice is specific for domain 13 and 14, as you can see here in this Western blot, where the sera recognizes full-length 230 from parasite material, but not 230 from the transgenic parasite line that lacks these domains. And similar in IFAs, the immune sera recognizes late-stage gametocytes and activated female gametes from wild-type parasites, but not gametocytes of the transgenic line lacking these domains. We also performed standard membrane feeding assays using the immune sera from these mice. In 2 experiments, we used heat inactivated sera, while in the 3rd experiment, complement remained active. And basically in all 3 experiments, we found that the sera of the short construct reduced the oocyst count significantly, while that of the long construct had basically showed no reduction in transmission activity. So one possible explanation for this is that when we produce the 2 constructs recombinantly, we observe a lot more aggregated protein for the long construct compared to the short construct, which potentially affects how the protein is presented, causing an antibody response to different and non-transmission blocking epitopes. So moving forward, we will focus on a short construct and we'll use protein design to further improve expression. So I hope I could show you that we're able to solve our cryo-EM structure of the 230, 48, 45 fertilization complex purified from endogenous parasite material. And from this, we gained a deeper understanding of how these proteins interact with each other and identified 230 domain 13, 14 as a new transmission blocking immunogen. So in the past couple of years, also others have now shown that domains other than domain 1 of 230 are targets of transmission blocking activity. And yeah, it's quite exciting that the field is moving forward to investigate these other domains. with this, I would like to thank a lot of people that were really important for this project across WeHi. I would like to thank my PI, Hong, for letting me work on such a challenging project, although I had no prior knowledge in parasite culturing or cryo-EM. Legion is a postdoc in the lab, and she established and performed all the pull-down assays. Jill generated a transgenic parasite line where domain 13 and 14 are missing. And yeah, all the beautiful IFA studies were performed by Jill. Frankie worked on the 48, 45 nanobodies and the bispecific nanobodies. Amy generated all our nanobodies. Lynn helped with nanobody purification, and she helped me a lot with the crystal structure determination. Mika generated the transgenic parasite line with the purification tech. Sash taught me how to culture parasites. Andrew taught me everything from cryo-EM crit preparation and microscope usage. Shabi and Eliza were very helpful and provided lots of guidance and help with data processing. Kathleen and Quentin for the generation of nanologics to analyze next generation sequencing of nanobody outputs. Ivo and Ria for the immune cohort study. And I want to thank many facilities across Melbourne, the mRNA Core, Australian Synchrotron, Eindhoven's Imaging Center, and the Tham Lab. And I want to thank CCE MMP for the opportunity to talk today. And with this, I'm done, and I'm open to answer any questions.
Yeah, thank you very much, Melanie, for the excellent talk and providing the complete picture right from a start of the complex purification to the preclinical studies. The floor is open for the question. Please unmute yourself and ask any question if you have. I think I should start with the first question till the rest will jump on. So, how do you think these nanobodies will be used as a either as a vaccine candidate or as a biologics to prevent the transmission of malaria?
Yeah, I see them in basically two different applications. So they could be used in passive immunization strategies, or as they're currently under development in these transgenic mosquitoes. That are developed by our collaboration partner, Dina.
Yeah, great. And how do you think that those antibodies which you have mentioned, the human antibodies, how, because the whole complex is a membrane-bound GPI anchor complex, yeah?
Mhm.
So, so how do you think that how the approach angle of these antibodies will affect the neutralization potential?
Yeah. I haven't shown the data on the nanobodies that are actually targeting domain 13 and 14 and have to compete with 48, 45. They are in standard membrane feeding assays that do reduce oocyst number, but not as efficient as other known antibodies. So they might be interesting in combination with nanobodies or antibodies against other domains or other antigens. But on their own, they are probably not the most promising candidates.
And are there any mutation happening in the malaria where they can uh, cause the resistance against these antibodies?
Hmm. So in general, the mosquito stage antigens are not that polymorphic because they are not under immune pressure in the human immune system. There are some sequence variations, but in the domains 13 and 14, yeah, they are fairly well conserved. So the
That's
epitopes of those nanobodies, are pretty conserved from different field isolates, if that answers your question.
okay.
Can
Yeah,
I just, oh, yeah, can I just ask a quick question? I'm fascinated by this idea of a transgenic mosquito,
Mm,
but obviously, I mean, this for me feels like, uh, I mean, I guess I got lots of questions. First of all, um, I mean, it's a big risk. Um, letting that out into the wild? How is that risk being mitigated? And also
mhm.
and I guess this speaks to some of Inamu's questions too, is long-term efficacy, consequences of evolution or co-evolution, some of these sort of things. Did you have some sort of, I guess, that wasn't really a question. I'm sorry,
Mm.
Melanie, that wasn't really a question,
No,
but I
no,
think you get the vibe of what I'm going for, yeah.
yeah, there are lots of ethical considerations of using transgenic mosquitoes in in the field basically. So they do a lot of so it's part of the transmission zero program, and they do a lot of comprehensive risk assessment to judge the environmental impacts. And they do a lot of community consultations to have like the support of the local communities, which is quite an important factor. So they are
Yeah, because I mean, I know in Australia we've been running the Wolbachia program for dengue virus and Ross River and a few of these ones. But I mean, this seems like, I mean, in my opinion, seems like a little bit of a less of a risk because you've got a natural sort of bacterial infection that these guys pass along. There's anyway going to be natural co-evolution as a consequence of that. But yeah, I guess it just that idea that if you provide a selection pressure that's like really strong within a population, what are the long-term consequences for evolution of more resistant strains of Plasmodium and all those. Yeah, I guess this is maybe beyond the scope of this talk, but I guess I heard this and a bunch of alarm bells started flashing in my head. But obviously, this is not my field, and I don't know much about it. But yeah.
Mm, yeah, it's a very I think it's a very interesting concept, but it's as you said, there are a lot of alarm bells like, oh, is this actually a good idea? But they definitely investigate the mosquitoes. They don't apply the gene drive right away. So they first evaluate the transgenic introduction first on a different level.
so I think we have a very nice discussion. And are there anybody who are willing to jump in and ask questions? Oh, okay. That's great talk and great presentation. And do you think that I'm just one more question from the maths discussion? So